Hello everyone. What we're going to talk about in this lecture is, if you want, one example of what I've anticipated when we were talking about buffers and when we were talking about the importance of the hydronium ion dissolved in aqueous solution on the structure of biomolecules. So here we'll talk about how the pH, so the concentration of the hydronium ion, affects ultimately the charge and the behavior of amino acid. And then at the end we'll talk also about how you can exploit the properties of the amino acid at different pHs also to separate them. So first what we'll talk about, we'll review the structure and the classification of amino acids, and then we move onto the properties of amino acid. First, we'll briefly talk about their polarity. We'll see how they are all polar but with different gradients of polarity, and how this property can be exploited to separate them by using a technique like thin layer chromatography. And then we'll move into the relationship between the pH and the charge of any amino acid, and how this can be used to separate them by using ion exchange chromatography. So, reviewing the structure of amino acid. I'm sure you remember all of this from your first biology, but all amino acids, they do share common properties, which are the presence of a central carbon, which is referred to as the alpha carbon, which then has bound to it for different groups or elements. So you have always an alpha amino group attached to it. The alpha amino group is called alpha to distinguish it from any other amino group that might be found on the R chain here. Then you have a hydrogen, an alpha carboxy group, again named alpha to still distinguish from any other carboxy group possibly found on the fourth group here, which is found attached to any amino acid, which is unique for a specific amino acid. All together in nature you have 20 we have 20 amino acids, and therefore there are 20 different types of different R chain here, which is what gives them the properties and basically makes them classified. I want to stress one thing: when we go through classification of amino acid, it's all based on the R group. You'll see, so amino acids are defined as polar and non-polar, but that's all based on the R group, and you see that in reality they are all polar later on. Another important thing to remember is how amino acid, the same amino acid actually can exist because of the alpha carbon here, can actually have this group arranged in different ways. And so the same amino acid can actually exist as different stereoisomers. What do we mean by stereoisomers? Are type of isomers that have therefore the same formula. Obviously it's the same amino acid, so it has the same formula, the atoms are attached in the same way, in the same organization overall, as you can see here in these two forms of the same amino acid here. But they might have different three-dimensional organization. So having different three-dimensional organization here within these isomers makes them stereoisomers. Now, stereoisomer can be chiral or achiral, and all except glycine there can actually be chiral, the amino acid. And by chiral, what we mean is that once you have that the organization, three-dimensional organization of the groups attached around the alpha carbon in the same amino acid and gives origin to two amino acids which are mirror image of each other, therefore these will be called chiral stereoisomers. Okay, and so this identifies a D or dexter or Leber's version of the same amino acid again, which are chiral stereoisomer of each other here. One example, if you're still unclear what chirality means, and chiral comes actually from the Greek word chiral, which is means hand. And the reason being that if you compare your two hands, you will see that they actually look exactly like a mirror image of each other. Whereas if you, for example, get these two glasses of wine, they look basically identical to each other, but is independent from their arrangement, and they are considered a chiral. Or if they could not overlap with each other, they will be considered achiral. So amino acids do exist. The same amino acid can exist in different stereoisomer forms, which are chiral. So mirror image to each other. And now these determine a D and a L, or Dexter or Lewis version of the amino acid, and DLL is determined by similarity to glyceraldehyde, so there is not a specific rule for that. And this leads ultimately to the existence of these two forms. They are different. Biologically speaking, these are two different forms of the same amino acid. And in protein, you always find the L form of amino acid. So only one of the two chiral stereoisomer for the same amino acid. As I mentioned before, amino acids are classified based on their R groups. This is an artificial classification, right? When it comes particularly to dividing them between polar and non-polar amino acids. All amino acids are polar because they all have an amino group and a carboxy group, right? The alpha amino group and alpha carboxy group. But based exclusively on the R group, they can be distinguished between non-polar and polar. And then the subgroup, the polar subgroup, can be divided further into uncharged or charged amino acid, and the charged group can be divided into acidic or basic amino acid. Now, when we talk about amino acid, they can all be referred with their full name or sometimes with a 3-letter or 1-letter abbreviation. When it comes to the 3-letter, usually it's the first 3 letters unless these 3 letters are already taken. For example, glutamic or glutamate or glutamic acid here as the 3-letter GLU. However, when it comes to glutamine, which would have GLU as well, since the 3 letters are already taken, now you have the the 3rd letter is replaced with another letter there. The same thing for the first letter, might be available or not. So with glycine, you have the first letter G. Glutamate and glutamine, the G is already taken, therefore the first letter is different from their letter, single letter is different than their first letter. So in this case, E for glutamate and Q for glutamine. Now, let's go through the different groups and the amino acids that belong to them. Let's start with non-polar. In total, you have 9 out of 20 amino acids which are non-polar. There are 5 here and 4 in the next slide. So, and they all have a classic R group, which is either made of a simple hydrocarbon like alanine, or a bit more complex one like a branch one like in leucine, proline, valine here. Glycine being the simplest one, which doesn't even have a hydrocarbon here, has only hydrogen. But all of them, as you can see, being based in this case on hydrocarbons, they are all non-polar. Then you have these 4 other amino acids here: methionine, tryptophan, phenylalanine, isoleucines. They may have some ring here, hydrocarbon ring, and other elements which, though altogether, don't really change the polarity of the R group. So all of them have non-polar R groups, and therefore they are classified as non-polar amino acids. The next group is polar but uncharged amino acid. They are defined as uncharged because even though they have some polar group, like this hydroxy group, this thiol group here, hydroxyl group here, and so on, these groups do never ionize. What I mean by that is that they never lose, for example, the hydroxyl group here never lose the proton here. Same thing for this thiol group, and so on. This group here doesn't never accept a proton. So by never accepting or releasing a proton or hydronium ion, they never acquire a charge. They always stay neutral. So they are polar but neutral, so uncharged. Then there are 5 other amino acids which instead can ionize. 2 of them are classified as polar charged acidic, which are aspartic acid and glutamic acid. As you can see, they all have this carboxy group, which, as you know, acts as an acid. Can exist as COOH or lose the proton and therefore exist as COO minus, so acquires a negative charge, hence charged acidic amino acids. The opposite for the basic ones, which have groups like this amino group here, for example, that can accept a proton, acts like a base, right? Can accept a proton, and therefore they're polar and they become charged, so polar charged basic as classified. So that's lysine, histidine, and arginine. Now, We have reviewed the structure of amino acid. Let's move into their property. Let's talk first about their polarity very briefly. So, as I just mentioned, they are classified as polar or non-polar. That's the first big distinction between amino acids. However, as I stressed before, they are all polar. So don't get deceived by that definition, which is based only on the R group. However, they have different degrees of polarity, so which is based ultimately on the properties of the R group. So if they have a very long hydrocarbon, for example, like this one, it will be less polar than one that has a shorter one, like this one. And obviously, if you start having some polar groups attached to that, then at this point the polarity will be increasing. So all amino acids, they're polar, but they all have different degrees of polarity, which is what is exploited in thin layer chromatography. Sorry, there's a mistake here. It's meant to be thin layer chromatography here, which separates the molecule, and you do that in the lab exclusively based on their degree of polarity. How does thin layer chromatography works? Well, in general, for any molecule that is polar can be separated by thin layer chromatography. And this is done by putting what we call a stationary phase, which is nothing else than a plate which is coated with some polar molecule. Like in your case, you'll be using a plate that is coated with silica, which is polar. And then a mobile phase, which is a buffer made of different combination of different molecules, and the ratio of the different combination can give it more or less polarity. So this buffer, so the plate is immersed inside a buffer, and the buffer, with a certain degree of polarity, will move by capillary action through the plate. Therefore, any molecule that is put on the plate, spotted on the plate, will find itself spending some time with either the stationary phase, so bound to the stationary phase, or following the mobile phase, the buffer, in its movement upward by capillary action. Now, the buffer can be more or less polar compared to the plate. So, if it's higher, so more polar than the stationary plate, you talk about a reverse phase chromatography. If it's less polar than the stationary phase, so then the plate, or the silica on the plate in this example, it will be referred to as a normal phase chromatography. One example of a solvent that you can use in the lab is the one you'll use in your own lab, which is a mixture of isopropanol, acetic acid, and water, or PaW solvent. With different ratio, you can achieve different degrees of polarity there. So now, how does this lead to separation of molecules based on their degree of polarity, such as amino acid? Well, now, depending on the polarity, so now you know there is a phase, a stationary phase with a certain degree of polarity. It's assumed made of silica. And then you have a polar buffer, which may be more or less polar compared to the stationary phase, moving on top of the stationary phase by capillary action. So now, any molecule that is put on the plate, so imagine you have this mixture of these two amino acids there, A and B, put on this plate. So right now they are stuck to the stationary phase. You just put them there, there is no buffer yet. But then, let's assume that you're doing a reverse phase chromatography, so you're using a mobile phase which is more polar than the stationary phase. And now this phase start moving through the silica plate upward. At a certain point, it will go through the molecules that you have plated there. By doing that, now you would have that molecule A and B will start spending some time attached to the silica plate, so the stationary phase, and sometimes with the solvent, the mobile phase. Now the time that they spend, the relative amount they spend with the stationary phase compared to the solvent, depend on their degree of polarity. So in this case, molecule A is less polar than molecule B, and therefore this molecule will tend to spend a little bit more time with the less polar phase, which is the stationary phase. Whereas the molecule B will do the opposite, will tend to spend a little bit more time with the more polar phase, which is the mobile phase. Therefore, as a result, this molecule A will stick hyphenated a little bit more to the plate compared to molecule B. And when the buffer is moving through that will move molecule B farther than molecule A, hence causing the separation of these two molecules. This can be used as it will be used in the lab also for identification purposes, and I'll leave it to your instructor to explain how you can use this for that purpose. But the principle, the migration is still the same based on the degree of polarity. Let's move on now to the second property of amino acid, which is the charge, which changes in relations to the pH of the solution. And again, this is the example on how water with the ion, in this case the hydrogen ion present in solution, has a huge effect on the property of molecule, in this case amino acid. So now, any amino acid, because of the alpha amino group and the alpha carboxy group, could ionize, all right? So could lose or accept a proton, right? In the case, and because of this, now each of this group will be acting like a base or an acid, for example. And therefore, amino acids can be considered as polyprotic acids, right? They are for sure acids. So this group can bind one proton, this group can bind another proton. So release a proton, sorry, this can release a proton, so it can be considered a polyprotic acid. Like any polyprotic acid, therefore, they would have one pKa for each ionizable group. And so they would have at least two pKa's, one associated with the amino group, which in this example is 9.6, and the other one associated with the carboxyl group, which is 2.34. Now, this, I hope you can see, it dictates also some change in charge. So imagine if you are extremely low pH. Now, at this point, you would have that both groups will be protonated, and therefore the overall amino acid would have a positive charge. Now, the higher the pH goes, the more these groups start deprotonating, with one of them, in this case the carboxyl group, will start deprotonating first, acquire a negative charge, therefore the molecule will become actually neutral. And then if you keep going, even the amino group will start losing the proton, becoming neutral. And therefore the overall amino acid, this example is glycine, the simplest one, will become negatively charged. But I hope you can start seeing how the charge of the amino acid is highly dependent on the pH. It's completely dependent on the pH of the solution is found in. If you put it in a solution with a very high alkaline pH, you would have a negatively charged amino acid. Put a very low pH, you would have a positively charged one, and you can go transition through the neutral form as well. Now, so all amino acids have at least two ionizable group, which are the alpha carboxy and alpha amino group. Some amino acid, the one you see right here, arginine, aspartic acid, cysteine, glutamic acid, and so on, those that are either acidic or basic, you remember the charged amino acid, will have also a third ionizable group present in the R group. So in this case, for example, this is glutamic acid. It has this additional carboxyl group, which has also its own pKa. Now, the values in terms of pKa for each of these groups can be found easily on different tables. You can find them on the web, but for example, you can use this one right here, which tells me, for example, alanine has only an alpha carboxyl group, alpha amino group, and therefore only two pKa values. Whereas if you move to arginine, now it has the classic alpha carboxyl group pKa values, and one for the alpha amino group, which is 9.04, but now you start having also a alkaline group here, a basic group here on the R side with a pKa of 12.48. So for any calculation that we'll do later on, if you're looking for information about pKa, you will get them straight from a table like this one. Now, because of the existence of two or three pKa's, amino acid act exactly like polyprotic acid, and therefore would have a number of buffering range corresponding to the number of pKa's that they have. So if they have only two pKa's, the one for the alpha carboxy group and one for the alpha amino group, they will show two buffering range. If they have an additional R group which can be ionized, they would have a third buffering range. And you can see that when you are titrating them. So assume that you take, for example, an amino acid and you start at extremely low pH, with what is called the hydrochloric version of the amino acid. That's what you'll do if you're doing the titration of the amino acid. At this point, it's fully protonated, and then by adding a strong base, you will go through all the different um, buffering ranges and therefore the different pKa's there. Now, titrating a amino acid could actually allow you to identify. You can allow it to identify it in different ways. One of them is by analyzing where the buffering ranges and the pKa values are. So if you can see, you know, by definition, a buffering range is 2 pH units, right? So which goes between pKa1 and pKa plus 1 and pKa minus 1. So for each group, for example, going back here, if you're talking about alanine, you would expect to have a buffering range between 1.34 to 3.34 and another one between 8.69 to 10.69. And so if you don't know what amino acid you have and you look at the buffering ranges there, so here already know that there are 3 buffering range. Based on where they are, I know that in the middle point there are the pKa values. And so I can actually identify also the pK values of this amino acid and hence deduct, based on the knowledge here, which amino acid it could be. Together with that, you can also use the the equivalence point, and this is something that will be explained to you by your instructor in the lab. So all of them have different buffering range corresponding to the pKa of each group that they have, plus minus 1. Great. Now, let's start introducing the relationship between the charge and the pH. So now you know that if you are titrating an amino acid, so you take your amino acid and you start adding either a strong acid or a strong base, you will go through these different plateaus here. At the same time, you actually be ionizing and deionizing, so adding or removing protons from any of these 3 groups. Right? Now, if you start with a hydrochloric version of an amino acid, so something that's been heavily treated with hydrochloric acid and therefore fully protonated, now you can imagine you will be going from a positive charge because all the groups will be protonated, including the alpha amino group, which would have a positive charge, eventually the additional one too. And then if you keep removing them with an hydroxyl ion, you will slowly move from a positive version of the amino acid to a negative version of the amino acid. In between, you will go through a neutral form of the amino acid, which is referred to as the zwitterion, okay, which is obviously between the positive and the negative charge. I'm going to show you in the next slide titration. So now the pH value during the titration that brings all the amino acid, 100% of it, in the neutral form. So once you reach that value, for example 7.38, right, pH value that gives you 100% of your amino acid all in the neutral form, that pH value is referred to the isoelectric point. And this is different from the equivalence point by definition. The values might overlap between the isoelectric point and the equivalence point, but by definition they are defined very differently. The equivalence point is the point in the titration where you have added an equal amount of titrant. So if you're using sodium hydroxide, an equal amount of sodium hydroxide to the molecule that you are titrating. In this case will be the amino acid. Once you have added the same number of moles, you have reached the equivalence point, and that pH value will be your equivalence point. Whereas the isoelectric point has nothing to see with that, is that pH value reached when 100% of the molecule is in the neutral form. Let's give you some example here. In this example, this is an example of a titration of glycine. In this case, actually the isoelectric point and the equivalence point, they do correspond. So imagine here you're starting with the hydrochloric version of your amino acid, so fully protonated, heavily treated with hydrochloric acid, so fully protonated here. So all the groups In this case, glycine has two groups and therefore two pKa's, the alpha carboxyl group, alpha amino group. Both of them are in the protonated form. The alpha carboxyl group is in the neutral form. The alpha amino group is in the positive form. Then you start adding some equivalents of hydroxyl ion. At this point here, you will go first through the first pKa, which is the one referring to the hydroxyl group. Has a lower value compared to the one, so the carboxyl group has a lower value compared to the one of the amino group. So the pKa relative to the alpha carboxyl group for glycine, if you go back to the table, you will find it there, has a value of 2.34, whereas the one for the amino group it has a value of 9.60. This tells me that during the titration, you will go first through the deprotonation of the carboxyl group, which slowly will convert from the neutral form into the negative form. And then you will go through the one of the amino group, which then will lose the proton and go from positively charged to negatively charged. So you're adding equivalents of sodium hydroxide. Therefore, first you go, you are deprotonated in this group. This is acting like an acid. If you remember, based on the Anderson and Hasselbalch equation, once you reach a pH which is equal to the pKa, in this case of the carboxyl group, you will know that you are at a point where you have 50% of this molecule in this form, so 50% of the carboxyl group in the protonated form and 50% in the deprotonated form, right? That's what gives you pH equal to pK. And then you slowly start having more of the amino acid with the deprotonated carboxyl group compared to the one protonated. Once you get to pK plus 1, the end of the buffering range, you will have 10 times more of this form compared to this one. If you don't remember, go back to the buffer, to the lecture on water, pH, and buffers. At a certain point, though, you will have fully deprotonated the carboxyl group. The amino group has not been deprotonated yet. And you will have not started deprotonating the amino group yet. At this point, once you have completely deprotonated the carboxyl group, but you have not started deprotonating the amino group here, you would have that this group is in the negative form. In all molecule, 100% of your amino acid will be in this form, with 100% of the carboxyl group in the negative form, 100% in the positive form. At this point, all of your amino acid in this solution, at this specific pH you're going through, will be in the Zwitterian neutral form. Hence, this is your isoelectric point. For glycine, this will correspond also to the equivalence point, when you have added an equal amount of hydroxyl ion to the amount, to the moles ultimately, of amino acid that you have in the solution. But this is a coincidence. By definition, they are different things. Okay. After that, excuse me, if you keep titrating with hydroxyl ion, keeps removing protons here, and therefore now the alpha amino group will start deprotonating. Once you have 10 times more molecules in this form compared to this, you're reaching the pK2 minus 1 point, the beginning of the buffering region associated with this group acting as an acid. Once you reach the pK2, so 9.6, at least theoretically, you should be at a point where you have 50% of the amino group in the protonated form and 50% in the deprotonated form, and therefore 50% of the amino acid in this form, 50 in this form here. And then you will move on. You get to the pK2 plus 1 point, the end of the buffering region here, where you would have 10 times more of the molecule at this point in the negative form than the one in the neutral form. And then if you keep going, 100% of the molecule will become negative. This is to show you, first of all, what we mean by titration of an amino acid, how it shows you the pK values in form of the buffering range with the value in the middle there, to show you where the isoelectric point is, or the equivalence point is, at least in this case for glycine they coincide, and what it corresponds to, the 100% of the amino acid in the neutral form. This is also to show you how, depending on the pH or where the solution is, you may have your amino acid mostly or completely in the positive form, or in the neutral form, or the negative form. So the relationship between the pH and the charge of the amino acid. These apply to any amino acid, the difference being that if they have a third pK values, then you would have a third buffering range in between. Now, again, I want to stress on the difference between the isoelectric point and the equivalence point. This is something that is always confusing for some of you. Now, the equivalence point, again, by definition, is the value that you reach when the number of the mole of titrant here is equal to the number of mole of the molecule you are titrating, so the amino acid right here. Once you have added exactly the same amount, you reach the equivalence point. Whereas, and this is always between pK1 and pK2. You'll see in the next example too. Always between these two pK. So if you average them, actually you can predict it. The isoelectric point instead is that pH value when you reach all 100% of the molecule, in this case the amino acid, in the deuterium form. Again, the value might coincide, like in the case of glycine, to the equivalence point, but by definition they're different. And let's go through some examples to show you that. For example, lysine here. These are the values that I've taken from the other table before, relative to the pK values of the three groups that you find in lysine. So the alpha carboxy group, this group here, has a pK value of 2.18. The alpha amino group has a pK value of 8.95. And the amino group on the side chain, on the R group here, has a pK value of 10.79. Okay. Now, let's assume for this example you're starting with 100 molecules of amino acid. All right, that's a ridiculous example, but just to give you numbers. Imagine you have 100 of this molecule in the solution. Now, practically speaking, means also that you would have 100 of this group present there, 100 of these alpha amino group present there, and 100 of these R amino groups, so the amino group in the R chain of lysine. All right, so that's what you're going to be deprotonating throughout. So you will have 300 protons that are going to be removed through the different step of the titration. And let's assume you're starting with the fully protonated hydrochloric version of your amino acid like this. So now let's see the relationship between the different charges that you get, the different stages, and the moles of sodium hydroxide that you will be adding. So you're starting at the beginning, no sodium hydroxide is fully protonated. All groups have their protons in there. Now, what is telling me this information here is telling me that the first group to be deprotonated here will be the alpha carboxy group because it has the lowest volume. Then the alpha amino group will be the second one, and then the one on the side chain. Keep in mind sometimes these two values may be flipped for other amino acid. Okay, so it's not always in this order. So let's start titrating. Imagine you start adding sodium hydroxide. So the first group again to deprotonate is the alpha carboxy group. So what's going to happen? Once you start having added 50 molecules, this is not moles but molecules of sodium hydroxide, now you would have added half of what you have in terms of amino acid in that solution. Therefore, half of the number of carboxy group that you have in there. So if you have added half of this hydroxy group, you can predict that you would have now at this point 50%. Let me change that with a pen. At this point, it would have changed it. So 50 molecules would have the carboxy group still protonated, and 50 of them would have the one deprotonated, which is exactly what the pK1 is, right? Is the pH is equal to the pK when you have 50-50. So same ratio, 1-to-1 ratio between the, if you want, conjugated base and the weak acid here. Right? Now, if you keep going at a certain point, once you have added the same amount here, then this, you would have fully deprotonated this group. So all your amino acid at this point will be in this form, with the fully deprotonated carboxy group and fully protonated alpha amino group and R amino group. All right. At this point, you would have added the same amount of titrant, right, than the molecule of amino acid that you have there. So the pH that you will reach here, this will be referred to as the equivalence point. So at this point, when you reach equivalence point, you have just deprotonated the first group. Right? The other groups are still deprotonated, and then you keep adding. And so the next group to be deprotonated at this point will be the alpha amino group, right? Based on the value I have here. So once you reach the pK2 at this point, just going a bit faster. Oh, actually, so once you reach pK2, you will be at 50-50, so you would have 50 and 50, right, of this group, 50% or 50 molecules at this point, which are still protonated, and 50 of the alpha amino group, which are already deprotonated. That's when you have 150 molecules of cellular hydroxide, so 1.5 times of the molecule of amino acid. And once you have added 200 of them, at this point you would have had fully deprotonated also these alpha amino group here. And now your molecule will be all in this form right here. And then if you keep going, now the third group to start being deprotonated will be the R amino group here, which has a pK of 10.79. So once you reach 10.79 at this pH, oh sorry, I forgot to mention that here at this point, excuse me, if you look at the overall charge, that was the point of this slide here. If you look now at the overall charge, you have one negative charge, one positive. The overall charge here is neutral. Therefore, the pH that you reach at this point right here, this pH value will correspond to the isoelectric point. You can see how the equivalence point and the isoelectric point for lysine will actually be reached at different point. The pH value corresponding to the equivalence point will be different than the one when you reach the isoelectric point. Then if you keep moving on, then you will start deprotonating this group. Once you have added 3 times as many molecules compared to the molecule of amino acid, you would have fully deprotonated your amino acid. All right. Again, this to stress on the difference between the equivalence point and the isoelectric point. By definition, sometimes the value might coincide, like for glycine, right? The example here. So in this case, the equivalence electric point, by definition, they're different, but the value, the pH values are the same. This is both the equivalence point and the isoelectric point for glycine. When it comes for lysine, we just went through, the isoelectric point is this pH value that you reach at this point right here. This will be your isoelectric point, whereas your equivalence point will be the one that you reach at this point right here. So something like that. Sorry, not perfect, but that's your equivalence point. So they are different. Sometimes, for some groups, like glutamic acid or aspartic acid, obviously because of the carboxyl group that starts from a neutral, not a charged position, and then and loses the proton afterwards. In this case, the equivalence point and isoelectric point will coincide too. But again, by definition, they're two completely different things. The value might or might not coincide. Now, you can actually predict, calculate where you would expect to get the isoelectric point for each of these amino acids based on the predicted titration that you will get with the amino acid. So for any amino acid, what we have done so far, we always started with the fully protonated version. So if I give you, for example, an amino acid and I ask you to predict where the deuterium will come out. So first you write it all completely protonated, right? So in this case, this will be lysine completely protonated with carboxyl group, amino group here protonated. If you count the total charge, see that this has 2 positive charge. Then once you start protonating, you will lose 1 proton at a time, and therefore this will become first +1. This form here will become neutral, so that's where the deuterium is, and then it will become negatively charged. When it comes with glycine here, if you look at the fully protonated form, you know, has a 1 positive charge, so it will become immediately neutral after the first group is completely deprotonated, and then will become negatively charged after the second group is deprotonated. For glutamic acid, same thing. If you fully protonate this, you have only one positive charge here, so +1 become neutral. Then will become -1 and -2, and so on. So now you can actually predict as a number where the pi or isoelectric point will be by just calculating the average of the two pKs that do contain the zwitterium. So for lysine, you can predict that the pi will be equal to pK2 plus pK3, whatever value they are, you get them from the table, right? And you know what it corresponds to, divided by 2. For glycine will be same pK1 plus pK2 divided by 2. Same thing for glutamic acid will be pK1 plus pK2 divided by 2, because that's the one that contains the zwitterium. Now, the equivalence point instead, where you add an equal amount, you have added an equal amount of titranol, in this case hydroxyl ion, will always be the same, will always be between pK1 and pK2. Notice here, this is the equivalence point right here. It's always between pK1 and pK2. So if you want to predict where the equivalence point is, you just average pK1, pK2. If you want to predict where the isoelectric point is, so the pH value where you will get 100% of your molecule in this deuterium form, first you figure out which pK values contain the deuterium, and then you average them out. And that's what I was telling here in this slide. And obviously all the values are given to you in different tables you can find online, or you can use this one, you can use that. So for example, here's when it comes to lysine. Now the two pK values of lysine right here, sorry, there are three of them, excuse me, 218, 8.95, 10.79. We have seen during the titration before that is between pK2 and pK3 that you have the deuterium form, and therefore you would average these two values out for the isoelectric point. For glutamic acid, for the isoelectric point instead, sorry, glutamic acid is here, it's messing you up a little bit, it will be between the first two pKs, so 219 and 425. Notice how the carboxyl group on the side chain deprotonized before the alpha amino group. Instead, when it comes to equivalence point, will always be the average of the first two pKs. So in the case of glutamic acid, it will be the same as the isoelectric point. In the case of lysine, instead, they will be different because of the deuterium located between pK2 and pK3. Now, these can be also predicted. So until now we talked about one amino acid, a single amino acid, but these can also be predicted for a peptide. Okay, you can have as many amino acids attached together. But for example, if you have a tripeptide here, like the one given here, glutamine, arginine, and methionine being joined together, you could actually calculate it the same way. So first thing you should have, obviously, exclude the groups that are involved in the peptide bond. So in this case, the peptide bond will be between these carboxy groups, so the pK for this group will be excluded, right? Same for this, involved in that bond, and the same for this one, right here. So that mess you up here, so we'll be left with this, and this will react with this, okay, and then you end up with this carboxy group at the end. So this was flipped, sorry, glutamine was flipped in this example. So you will be left, so once you draw your tripeptide, you will be left with a group associated with this amino group, and so on. And this will be discussed in class, how this translates into predicting the isoelectric point. So, and you do it exactly the same way that was done before here, but in this case, obviously, you would have more ionizable group. But if you find the starting charge of that tripeptide, let's assume you start with +4 for whatever ridiculous, you know, after one pK, you'll be a +3, second pK +2, and so on. And then you find where this deuterium is, you average that out, you get predicted the isoelectric point. We'll go through some calculation in class. Okay, let's finish this lecture up showing you why it's important to understand how the charge of the amino acid is dependent on the pH and how it can be exploited to separate them. So, by now you know that at different pH, amino acid would have different charge. And that's what ion exchange chromatography does. It separates amino acid in a mixture based on the charge that they have at certain pH, which is dependent on their isoelectric point. And this is done by taking the amino acids, putting them through a stationary phase. So you would have your amino acid, let's put one mixture of amino acid there. They would have different charges. Say this one is negative, positive, positive. You put them through a stationary phase, which is erasing here, which would have a group with a certain charge. Imagine this is negatively charged. You would have that now this amino acid, which is in the buffer, which is the mobile phase, will go through the stationary phase. It will not bind because it's negative. And whereas the other two, amino acid 2 and 3, which are positively charged, will bind to the resin, right? That's what ion exchange chromatography is doing. You run some buffer in a mobile phase, so in a buffer which gives them specific charges. You run them through a stationary phase which has a certain charge, and those that have opposite charge will bind, the other will move through. By doing that, you'll be able to separate one or more amino acid from the mixture. Now, how is this practically speaking? You plan the pH that you use for this amino acid in the buffer based on the pi. If you know the pi of a molecule, now you can really predict the charge that the same molecule would have at different pH. So if you put that molecule, so for example, glycine here, it's a bit off this, but it has a pi which is around 6, almost 6, a little bit lower than 6 when you do the calculation. But it's around 6. Take 6 as an example. Now you know that if you go at a pH which is lower than 6, all right? So if for the pi is 6 for glycine, if you go lower than 6, sorry, lower this way, than 6, now you can predict that glycine will be positively charged, which makes sense, right? If this is what you have, the pi here, anything at pH lower will be mostly negatively charged or fully negatively charged. Higher will be positively charged, most of it. The farther you go, the more obviously dramatic the difference is. So the higher, again, than 6, it will be negatively charged. This is valid for any amino acid. This is a simple example with glycine, but it's the same thing for lysine, glutamic acid, aspartic acid. If you predict their pi, remember by averaging the 2 pKa's that contain the zwitterion, and you put the molecule in a pH lower than that pi, it will be positively charged. Put it higher, it will be negatively charged. How does it help with ion exchange chromatography? How do you use it? Well, imagine, for example, you have these two molecules. You're given a mixture. So these two molecules, A and B, are exactly in the same test tube, right? One has a pi, you calculated, of 5.8, molecule A, and molecule B has a pi of 3. Now, you can actually predict the charge of this molecule exactly doing the reason I said before. So, if these two molecules are suspended in a buffer, right, lower than their pi, they would have a positive charge. So, if it's lower than 5.8 for molecule A, it will be positively charged. Lower than 3 will be positively charged molecule B. If it's higher, so higher than 5.8 here, will be negatively charged. And the same thing for B. Higher than 3 will be negatively charged. So I hope you can see now that basically if you choose a pH which is lower than the lowest pi, both of them would have the same charge, will be positively charged. Now there is a range in between the pi of the two molecules, right, where you're pretty much guaranteed, if you find it right in the middle point, right, let's say 4.5, you're pretty much guaranteed that you would have at this point one molecule, in this example molecule A, which will still be positively charged because 4.5, for example, is lower than 5.8. So we're positively charged. But now you know that you have manipulated at that pH molecule B, making it negatively charged. So now you have that these two molecules have opposite charges. Then if you go higher than 5.8, obviously both of them will become negatively charged. You can see, I hope you can see two things. You can plan a pH where you have molecules with opposite charge. Second thing, how you can actually change this molecule. So in this case, for example, A, if you move from a 4.5 to 7.5, you will move from positive to negative. All right, so during the same experiment. So how does this translate into separation? So well, imagine now that you are using as a stationary phase. So the stationary phase, just a few words here, correspond to the column here, which is filled with basically a resin that has attached a charge group. It's referred to as charge prosthetic group. And now this can be an anion exchanger. This is an example, DAE. It's called an anion exchanger because when you start, it's bound to chloride, which is negatively charged, so an anion. But then we'll replace the chloride with any other molecule which is negatively charged. So we'd exchange anions. It will bind to anions, basically, to negatively charged molecules. Or you can have a cation exchangers like DOX50, which is negatively charged, so it's bound original with sodium ion here, but it will replace it with any other positively or any other cations. All right, running through this. So imagine, for example, that you're working with DOX50, so positively charged. This is an anion exchanger. And now, sorry, a cation exchanger. And now you have this mixture. Now, to separate them, you could start with a pH, which is right in between here. For example, 4.5. What would happen in this scenario there? You would have that if you ran now this mixture at pH 4.5, molecule A, so amino acid A in this example, will be positively charged and therefore would bind with the negatively charged cation exchanger, the OX50. Whereas B is negatively charged, it will be repelled and it will proceed, will follow the movement of the mobile phase, the buffer, downward. And therefore, you would actually be able to, so at 4.5, you will be able to collect it in a sample separate from molecule A, which is still stuck, if you want, with this the stationary phase. Not only that, but then you can also move on and get also molecule A down. How do you get it to separate here is by changing the charge from positive to negative. And so what you would do, you will move to a pH higher than the PI. For example, 7.5, A will become negatively charged, and therefore if you keep moving, obviously you're keeping collecting sample there, you will get it to come out to a different test tube than molecule B, and therefore you would have achieved the separation of these two molecules based on their charge by playing with their relationship with pH and charge. So, summarizing all of this, what did we talk about in this lecture? Well, we talked about amino acid. Again, as one example of a biological molecule that is affected by the pH, ultimately, of an aqueous solution. So we first reviewed their structure. We talked about their common structure based on the presence of an alpha amino group, alpha carboxy group, a hydrogen, and then an R group. Then we talked about the concept of chirality, so how they are stereoisomers. So the same amino acid exists in two isomers, referred to as stereoisomers, which are also chiral, except for glycine, and therefore they are mirror image of each other. With the L form, there's a D and L form, with the L form being the one that is commonly found in protein. And then we went through the classification of the molecules based on the amino acid based on the R groups, polar, non-polar, charge, acidic, charge, basics. Then we went into the proteins of amino acid. We first talked about polarity, and we saw how all amino acids are ultimately polar, but with different degrees of polarity, and how this can be used by a technique like thin layer chromatography to actually separate amino acid. Then finally we talked about the charge. We talked about how the pH of the solution can affect the charge of the amino acid. Keep in mind that amino acid different charge are different biological molecules. Okay, and so we saw how amino acid will behave. If you look at a titration as well, you can see them how they behave as polyprotic acid, having multiple pKs, at least two, if not three, if they have a charge R group, and these with multiple buffering range as well. Then we discussed the definition of isoelectric and equivalence point, focusing particularly on the isoelectric point. How the isoelectric point is that pH value that you will reach in a solution when 100% of your molecules, so your amino acid, is in the neutral form. And then we discuss also the relationship between the isoelectric point and the pH of the solution. If you put an amino acid at a pH lower than the isoelectric point, its isoelectric point will be positively charged, higher will be negatively charged. And therefore, discuss how ultimately you can manipulate the charge of an amino acid, whether it's by itself or in a mixture, to be with a certain positive or negative charge depending on the pH that you put that solution at. And how these ultimately can be used to separate different amino acids by using ion exchange chromatography.